Now we are going to discuss the second part of this DNA replication mechanism, that is exposure of DNA strands and elongation. After that we will discuss the termination phase. So exposure of DNA strands means the two double helical structure, they are joined with the help of these hydrogen bonds. So this hydrogen bonds should be broken, and these are break with the help of the enzyme helicase. Okay, and suppose this is two strands are joined with the help of hydrogen bonds. When these hydrogen bonds break down, in that way they are open up, and this Y-shaped structure, this is called as replication fork. This Y-shaped structure is called replication fork, and single-stranded binding protein SSB bind with this single-stranded DNA so that it cannot again form this hydrogen bond or become double-stranded before replication process. Already we are discussing in my previous lectures about this SSB and regarding this helicase enzyme. Okay, and generally they are bind with this origin, and these are rich in AT rich region. Why AT? Already discussed in my previous lectures. So now after opening, now we move to this elongation phase. So in elongation, phase, suppose this double-stranded DNA, this is already suppose open up. So that means that is the replication fork part, and here already hydrogen bonds are there. Here hydrogen bonds break down with the help of these enzymes. Now already I told, now DNA polymerase play important role for this replication process, but DNA polymerase need a short segment of nucleotides. This is called as primer. Now suppose this strand is suppose 5 to 3, and this DNA strand is 3 to 5, and DNA synthesis occur which direction? 5 to 3 direction. So for polymerization, here RNA primer will bind, because that RNA primer will give the free OH group, and this is the 5' point, and DNA synthesis occur which direction? 5' to 3' direction. Now we are in which point? In 5 point. This is the primer, so this is the 5 prime, and here 3, that means here 3 OH groups are there. So now if here guanine is there, then the DNA polymerase can join cytosine. Okay, and that OH group, suppose here cytosine is there, or suppose here guanine is there, here in that primer position, then this nucleotide contain free OH group, and that nucleotide bind with this guanine with the help of phosphodiester bond, like that. In the direction, it is 5 to 3, it was synthesized continuously in this direction. Okay, but here now as it is 5 to 3, now the position is, this is 3. Okay, this position is 3. So here now according to the rule, DNA synthesis occurs which direction? 5 to 3, but here the position is 3. So how it occurs? Here primer bind, suppose this position, this is the primer. So here 5 to 3, this is the primer sequence. Okay, now with this 3 position, that means here OH group is there. So according to the complementary, DNA polymerase will join the nucleotides, and DNA synthesis occurs 5 to 3 direction according to the direction. does not break the rule. 5 to 3. Again, primer will join there, another primer. Again, it will synthesize in which direction? 5 to 3. 5 to 3. Now this is the primer position. Again, this is primer position 5. Then again 5 to 3, 5 to 3, and gradually it will open up and complete replication process completed. Okay. Here, this strand is synthesized discontinuously. So when it is synthesized discontinuously, when 5 to 3 act as a template. Template strand means the strand based on which the new DNA strand synthesized. In that case, 5 to 3, 5' to 3 strand act as a template. Then new DNA is synthesized, new strand synthesized discontinuously, or that strand also called as lagging strand. Okay, and these pieces of fragments, these are the DNA fragments. This is primer. This is primer. This is the DNA fragments. DNA fragments. Like that, this is primer. This is DNA fragments. That means they produce some fragments of DNA. These DNA fragments are called as Okazaki fragments. These DNA fragments are called as Okazaki fragments, as these are synthesized in pieces. In between, primers are there, so this strand is called lagging strand. When lagging strand form? When it take 5 to 3 as a template. Okay, but when it take 3 to 5 strand as a template, then new strand synthesize continuously. New strand synthesize continuously, and this strand is called as leading strand. This strand is called as leading strand. One strand is synthesized continuously, another strand synthesized discontinuously during replication process. But once they release from this replication mechanism or replication machinery, this discontinuous strand also become continuous. How? With the help of enzyme, that is RNase helicase activity. So that enzyme remove these RNA primers. And another one enzyme already we discussed in the protein parts, important protein parts, another enzyme that is called as ligase. That ligase enzyme finally ligates the DNA fragments, and that discontinuous strand also become continuous. Okay, that is the elongation process. Okay, another one last step is this termination. The termination, that means the DNA polymerase will be moved up to the end of the template strand. When it the end point, then the termination occur of DNA replication. That means as a whole, in exposure of DNA strands, hydrogen bonds break down, it will open up. So during elongation process, primers are bind and primers are synthesized primase enzyme. Okay, and when take 3 to 5 as a template strand, the new strand is synthesized continuously. From one single primer, it synthesized continuously because it is synthesized in which direction? 5 to 3. Okay, it will be hydrogen bond will be break, and again. Finally, like suppose from this, suppose this from 5 to 3, this is 3 to 5. Based on this, based on this strand, suppose 5 to 3, another one is 3 to 5. Based on this 5 to 3, this strand is synthesized. Like that, based on this, this strand is synthesized. That means from this strand, taking this as a template, new strand is synthesized. Like that, taking that strand as a template, these two daughter strands are produced during the replication mechanism. But when take 5 to 3 as a template, the new strand is synthesized discontinuously. Okay, but when they release from this replication machinery, with the help of this enzyme RNase H, it remove these primers, and finally ligase enzyme join these fragments, and this discontinuous strand also become continuous. And finally, when DNA polymerase reach at the end of DNA, so this strand, then termination occurs. Up to here, we have concluded the replication process in prokaryotes, already discussed in several lectures. So here, that is the end of the replication mechanism. Thank you.